some of our research for the past few years. And research in our lab focuses on understanding some of the factors underlying pathogen host shifts, largely using actually an insect virus model system, which given the scope of the meeting, I thought I'd sort of explain a little bit about, and I hope provide some sort of fundamental insights to help conceptualize a framework for understanding future disease emergence. So I don't need to tell this audience that a major source of emerging infectious diseases are host shifts, where a pathogen jumps from one host species to another. And we know that for a successful host shift to occur, the pathogen first has to come into contact with the novel host. It then has to replicate sufficiently to infect that new host, and then it must be transmitted at a sufficient rate if it's going to persist and spread in the novel host population. So here we're thinking about like R0 basically being greater than 1 for the pathogen to spread. These steps might involve the pathogen evolving and adapting to its new host, which in turn might allow it to infect further novel host species. And it's going to be this sort of middle step here that will be the main focus of my talk today. So first of all, I want to talk a little bit about pathogen virulence following a host shift. And we know that virulence following a host shift can be extremely high, as seen in these sort of high profile cases listed here. I should say I'm defining virulence here as the harm a pathogen does to its host. And classically, novel host parasite associations were thought to be the most virulent as the host and pathogen were mismatched, with long-term associations being thought to involve reduced levels of virulence. However, this has long since shown to not be the case. There's no empirical or theoretical evidence to support this, and many host shifts presumably go undetected due to the pathogen causing little or no virulence in the new host. And when we wrote a paper on this a few years ago, one of the reviewers actually suggested we take out the bit discussing this as to quote, no one believes the avirulence theory anymore. However, I think COVID-19 has shown that this view does still persist in the scientific and medical community. So I really think it's worth repeating that there's no evidence to support this idea of pathogens evolving towards avirulence. So at least when we carried out the study I'm about to talk about, there seemed to be no clear pattern as to what determines virulence following a host shift. Although I suspect we might hear a little bit more from my Lisa Fisher after the break on some of the neat sort of meta-analyses they've been doing on this topic, so I'll avoid any spoilers to their work here. So despite knowing relatively little about what determines virulence following a host shift, we know that the host phylogeny is an important determinant of a pathogen's ability to infect a novel host. And the first way this can act is through the effect of genetic distance from the pathogen's natural host. We know that parasites must either avoid or suppress an elaborate array of host immune responses and rely on their host for all their physiological needs. And this will result in them evolving specialized adaptations to their host. Therefore, parasites may be more likely to jump into species more closely related to the natural host, as they'll offer a more similar environment. And that's what's illustrated in this figure here. We've got a hypothetical host phylogeny. These bars represent some measure of parasite infection success, and the bar in red represents our natural host pathogen combination. And you can see that as we move into species more distantly related to the natural host, the pathogen doesn't do as well. So this is what I'll refer to as a distance effect. Oops, sorry, I'll skip forward there. However, related groups of hosts could also be expected to show similar levels of susceptibility independent of this effect of distance from the natural host. And this might mean that certain clades of related species could be particularly resistant or susceptible due to common ancestry. And I'll refer to these effects as phylogenetic effects. And that's illustrated here with this clade labeled B being highly susceptible despite being quite distantly related to the natural host. So we were interested to ask whether the host phylogeny is also important for determining pathogen virulence following a host shift. And to look at these questions, we carried out a large cross-infection experiment using 48 different species of fruit fly and a naturally occurring virus called Drosophila C virus, or DCV for short. DCV is a positive sense RNA virus that was isolated from Drosophila melanogaster, where it seems to be horizontally transmitted between individuals. In infected flies, their gut basically becomes blocked up. The flies sort of swell up a bit like a balloon and effectively die of constipation, so not a very nice way to go. But this is a really powerful model as it allows us to include a really wide diversity of host species and carry out experiments that would not be possible in a vertebrate system. So in this first experiment, we inoculated 48 species of flies by pricking them with a needle dipped in either the virus or a control solution. We then measured virulence in the flies, which is the mortality each day for 20 days post-infection. We measured the change in RNA viral load over a 2-day period using quantitative RT-PCR. And this is what the data look like here. On the y-axis, we've got the proportion of flies alive. On the x-axis, we've got day post-infection. I'm just showing you data for 3 different species here. Points in red are our virus-infected flies and points in blue are the controls. And you can see for this species here, the virus-infected flies die relatively quickly. Here they're dying, but at a somewhat slower rate. And in this species, they're hardly dying at all relative to the controls. When we look at all 48 species from the experiment, we see there's a great deal of heterogeneity in the amount of virulence we observe. So in total, we infected and measured the survival or viral load in about 12,500 flies over around 600 biological replicates. On the left here, I'm showing you the host phylogeny, and the common ancestor of these flies is estimated to be around 40 to 50 million years ago. If you want to tease any vertebrate biologists a little bit, we can suggest that given the rapid generation times and so high rates of evolution, these flies have sort of equivalent diversity to all mammals. On the right here, we've got the virulence data in blue. This is the mean proportion of flies dead. And in red, I've highlighted Drosophila melanogaster, which is the host that DCV was isolated from. You might be able to see that the different host clades vary in their degree of virulence, but this is perhaps rather hard to see. So what we can do is carry out ancestral trait reconstructions and plot virulence as a colour gradient across the tree. So here we've got a red being high levels of virulence. As we move to blue and green, we've got progressively lower levels of virulence. And we can use this to visualize how virulence changes across the phylogeny over time. So here we're looking at day 4 post-infection. Most of the virulence is localized to this clade down here at the bottom of the tree. As we move forward to day 6, not much changes. Day 8, we're starting to see virulence appear in this clade down here. If you keep an eye on that, as we move forward to day 10, suddenly the other species in this clade are showing increasing virulence. We're starting to see virulence creep up in these clades at the top part of the tree here as well. Day 12, we're now seeing high virulence in this sister clade, and again, virulence is creeping up here. Day 14, and by day 16 post-infection, almost everything's dead or dying, apart from species in this clade here. So it seems that the host phylogeny is something of a patchwork of different clades that vary in their degree of virulence. And briefly, I just want to say that we analysed this data using a phylogenetic mixed model. What this allows us to do is to include genetic distance from the natural host as a fixed effect, and then fit the phylogeny as a random effect. We can then fit this species term also as a random effect, and this includes any variation that's not accounted for by the host phylogeny, which means the model's robust to any deviations from a Brownian model of evolution. What this enables us to do is to calculate the proportion of variation that's explained by the phylogeny after accounting for this fixed effect of distance. And that's what I'll refer to as this sort of phylogenetic effect here. When we look at the results of this model, we firstly find there's no effect of distance from the natural host, which we've treated here as Drosophila melanogaster. This might be because DCV is something of a generalist pathogen. We know it at least infects the closely related Drosophila simulans in the wild, or it might be we're simply not correctly assigning the natural host. What we do find, however, is that the host phylogeny explains a large amount of the variation in virulence, as well as a large amount of the variation in viral load. This means that closely related species have similar levels of virulence or similar viral loads due to common ancestry. And such patterns may arise if the common ancestors of different host clades have either lost or gained immune or cellular components that affect susceptibility to DCV. We can also carry out ancestral trait reconstructions for viral load data, as seen on the right here, and you can see this mirrors the virulence data quite nicely. And if we look at a correlation between viral load here on the y-axis and virulence on the x, we find this very strong positive relationship. And given what we know about this system, this suggests that virulence is a direct consequence of virus accumulation in a host. So, continuing this work, we're interested in why we see these differences in susceptibility between different groups of host species. And I want to talk to you a bit about some work that was carried out by Kat Roberts, who's just finishing up a postdoc in a group here. So one thing that might be important is, we thought might be important, is temperature. And here I'm showing you some really nice data from Vanessa Kellerman and colleagues, where they measured the ability of flies to tolerate either low or high temperatures. And like virus susceptibility, there's phylogenetic signal in the thermal tolerances of different Drosophila species. And this has been linked to the distribution of these species in nature. So this means that different groups of hosts may have different thermal optimas. And so when we assay them at a single temperature, as we did in the previous experiment, the patterns we see might be due to some species being at suboptimal temperatures. This might mean that either the host immune response or the cellular machinery the virus hijacks isn't operating very efficiently. So we were interested in whether the susceptibilities of species respond in the same or different ways to changes in temperature. So basically we did the same experiment, but we kept the flies at 3 different temperatures. We had a low, a medium, and a high, which is 17, 22, and 27 degrees C. And then we measured their viral load. We also looked at the upper and lower thermal limits of these flies, as well as measuring basal metabolic rate across temperature. What we found is these very strong positive correlations in viral load across the different temperatures. However, when we increase the temperature, we find that the variance in viral load increases, but the means remain relatively constant. So typically when we think of an interaction across environments, we think of something like this, where we've got this sort of crossing of reaction norms. But for our data, the rank order of susceptibility of species remains relatively constant across the temperatures, hence these strong positive correlations. But as temperature increases, the most susceptible species become more susceptible and the least susceptible less so. I won't show it here, but we've got data that suggests this is unlikely to be due to the increasing temperature, increasing the rate at which infection progresses, i.e. basically altering where in the course of infection we've sampled. We also find strong phylogenetic signal in the lower and upper thermal limits of flies, consistent with previous studies, but we find these aren't significant predictors of viral load. We also measured basal metabolic rate across these temperatures. So super noisy, but again, doesn't seem to have a significant effect on viral load. So moving on, we were thinking about what other environmental variables could be important in understanding the patterns of susceptibility across host species. And Kat, who again carried out this work, has previously worked a lot on the importance of diet and understanding the outcomes of host pathogen interactions. And we know that DCV causes nutritional stress in the gut of flies, causing similar pathologies to those seen during starvation. So we wondered if, like we predicted for temperature, different species may have different optimal diets. And this could mean that for what for one species is a fantastic diet, this beautiful patisserie, for another, it's the equivalent of this, which if this was to appear in Bake-Off, I think a soggy bottom would be the least of its worries. So we know that the ratio of protein to carbohydrates in the diet are really important for a number of life history traits, including lifespan fecundity trade-offs, but also for pathogen resistance. So Kat placed 27 species of flies on 3 different diets with either a high, a medium, or a low protein to carbohydrate ratio. She then measured either survival following infection with DCV or control, or the change in RNA viral load. What we found was that changes in protein to carbohydrates in the diet did not alter the outcome of infection. We found strong positive interspecies correlations in both viral load and mortality across diets, with no evidence of a species by diet interaction. However, protein and carbohydrates are just one part of the diet. There's increasing evidence that lipids may be important, not just for virus resistance, but also for restoring the lifespan reducing effects of high protein diets. And so this may not be the end of the story here. This is something a master's student in the lab is currently investigating. So to summarize so far, it seems that these patterns of susceptibility across species are relatively robust to environmental perturbations. So we're sort of continuing to ask why some species are more susceptible than others. And there's some evidence that suggests that the tissue tropism of a virus can explain the severity of disease. Put another way, is virulence across species associated with infection in certain types of tissue? So we set out to examine this in just a mere 7 species of fly this time. And this was again work by Kat Roberts, who dissected flies up. So we broke them down into heads, which is pretty obvious, crops, and this is a food storage organ in flies. And a key symptom of DCV infection in Drosophila melanogaster is the crop sort of distending and swelling, which, as I mentioned before, ultimately leads to the flies dying of constipation. We've got the guts, we've got Malpighian tubules, which are the equivalent of insect kidneys. We know that infection is associated with water retention. We've also got sex tissues. All of these flies are male, so these are the testes and their accessory glands. And finally, we've got the body or cadaver, and this is basically everything that's left at the end. This is the cuticle and largely the fat body of the flies. And the fat body is involved in energy metabolism and immunity, and is a known early infection site of DCV. So here's what the dissections actually look like. For this photo, we put the flies on blue food to make everything stand out. These aren't actually Smurfs or eating Smurfs. So here we've got the crop. This leads down to the gut, which is nice and blue. Then here in these yellow things, the Malpighian tubules, so the insect kidneys, and these big swirly fellows at the top of the testes. We also then had the heads and the cadavers. We measured viral load in each of these tissues using quantitative RT-PCR. So in this figure, each of the different colours represents a different tissue, and each of the boxes here is a different species. The tissues are ordered by the rank order of viral load in Drosophila melanogaster at the top here. And what we find is there's significant effects of tissue type and of species, but no evidence of a tissue by species interaction. This suggests that in species with high susceptibility, this is due to increased overall susceptibility across all tissues, rather than the virus getting into and proliferating in a specific tissue. You can kind of see that here where these sort of species level effects are quite apparent. So we're currently looking at the virus's ability to bind, enter, and replicate in host cells, as well as its interaction with the host immune response across species. So far I've been talking about susceptibility to a single pathogen, but a key question is, are certain types of pathogen more likely to jump into some hosts, or is susceptibility specific to different pathogens? This is critical for understanding whether the characteristics of an emerging pathogen can be predicted based on our knowledge of other related pathogens. We might expect related pathogens to behave in similar ways, but we know that a relatively small number of mutations can have large effects on the virus's ability to infect a given host species, so perhaps this is not quite so clear. And we thought SARS-2 provides quite a good example of this, as when it first emerged, it was unclear if it would behave similarly to the closely related SARS, MERS, endemic coronaviruses responsible to common colds, or entirely differently to all. Several studies have highlighted the importance of virus taxonomy, basically virus genetics, in the likelihood of disease emergence. We wanted to try and test this experimentally. So this is some work by Ryan Imrie. Ryan's just finished up his PhD in the lab literally last week, and is now continuing with us as a postdoc. And Ryan has again infected lots of species of fly, but this time with 4 different viruses. We've got 3 different isolates of DCV, so we've got DCVC, EB, and M, as well as a more distantly related dysystrovirus called cricket paralysis virus. These different isolates of DCV were collected from Drosophila melanogaster stocks from around the world. Cricket paralysis virus was isolated from, as the name suggests, Australian bush crickets. The DCVs, as you can see from this phylogeny, are all quite closely related to one another, and they've got genome-wide nucleotide sequence identities of between sort of 94 and 97%. Cricket paralysis virus is more divergent from DCV, with the sequence identity between cricket paralysis virus and DCV being around 57%. These viruses cause different symptoms in Drosophila melanogaster. We know that DCV causes pathology in the gut, as I mentioned, whereas cricket paralysis virus doesn't seem to. Both suppress the antiviral RNAi immune response, but in different ways. So Ryan then produced this rather snazzy figure. So here we've got the phylogeny of our 45 different species of flies here on the left. Here we've got a cladogram, which representing the relationships between our 4 different virus isolates. And the bars here and the intensity of the colour represent the change in RNA viral load for each of the 4 viruses. The key thing we were interested in here was the correlations between the different viruses. So when we look at the correlations between the different DCV isolates, we find these really strong positive correlations, when correlations above 0.9 for all of them. When we look at the correlations between cryptoplastis virus and the different DCVs, we still do find significant positive correlations, but these are significantly weaker, coming in at around 0.57 on average. And this suggests that we might expect to see similar patterns of susceptibility to related pathogens, but unsurprisingly, this might drop off between more distantly related pathogens. The fact these correlations are less than 1 provides evidence for sort of virus by host species interactions with changes in the rank order of host susceptibility between the different viruses. That's illustrated in this figure here, where species are ordered by susceptibility to DCV-C at the top here. And we can see this order tends to break down as we move to more distantly related viruses, and we can see this shift in the rank order of susceptibility. So these findings suggest a sort of general rule of thumb is that the ability of a virus to infect a novel host can potentially be inferred based on relatedness between viruses, although we really need more viruses in a sort of study to say this definitively. Our data also suggests that host susceptibility is not specific to individual viruses, and that certain host clades may be broadly susceptible to infection by certain groups of pathogens. These susceptible host clades could potentially act as reservoirs or donors of emerging infectious diseases. And the next step here is to hopefully try this with a really broad range of different pathogen taxa that vary in their relatedness. And the hope is here we can kind of flip things around a bit and look at the importance of the pathogen phylogeny as well. So for the last couple of minutes, I want to talk about something slightly different. I've just talked a bit about what is effectively pre-standing genetic variation in a virus population and how this might be useful for understanding host shifts. We've long known that viruses often evolve and adapt to novel host species, as we're currently observing with SARS-CoV-2 and various variants. And I want to talk a bit about how viruses can evolve and adapt to novel hosts that vary in their relatedness. To do this, we carried out an experimental evolution study where we passaged DCV in 19 different host species, 10 passages. For each species, we've got between 6 and 10 independent replicate lineages of the virus, and in their final passage, we deep sequenced the genomes of these viruses. This is the phylogeny of our 19 different host species. I just want to highlight that these flies still shared a common ancestor around 40 million years ago. But what we've tried to do is include either pairs or groups of really closely related species to try and see if the virus evolves in similar ways in related hosts. This heat map is showing you the results of sequencing. Don't worry too much about seeing any of the detail here. All you need to know is that each of the rows represents an independent replicate lineage of the virus, and each column represents a position in the genome of DCV. The intensity of the shading represents a frequency of a mutation at that site in that specific lineage. And what we find is that parallel mutations, which are indicative of selection, are significantly more likely to occur either within a single host or between closely related host species. And this suggests that some mutations may allow a virus to adapt not only to the host it's in, but also to other closely related host species. So to wrap up, it seems that the host phylogeny is important in understanding a virus's ability to infect and cause virulence in a novel host, as well as being important for understanding how viruses evolve in new host species. Changes in temperature may alter the likelihood of host shifts into certain species, and we're just starting to understand how pathogen relatedness may be important for understanding the outcomes of host shifts. So that's all. I just need to acknowledge a few people I've already mentioned, Kat and Ryan, who carried out a lot of this work. the sort of stuff at the beginning, right at the beginning and the end, was done in collaboration with Frank Jiggins. Lots of other people also played a role, and thanks to Wellcome Trust and Royal Society for ongoing funding. So very happy to take any questions as long as there's time. Thank you.
Great, thanks, Ben. And we're running ahead of time, so we've actually got quite a bit of time, about 10 minutes for questions. So there's a couple of questions that have come in the chat. So I'm going to say this name probably completely wrong, but Isicio Ho from LSHTM asks, what are the factors that determine whether the cross-species transmission will result in spillover or sustained onwards infections?
Yeah, okay. So yeah, I mean, it's going to vary a little bit, but so we know for a lot of these, for DCV that we're studying on, it's something of an obligate killer. So it's going to be transmitted by replicating to high viral loads, and then we think the sort of natural route of transmission is actually larvae cannibalizing the corpses of adults, but that's sort of a bit of speculation. So, you know, as an obligate killer, it doesn't really matter how quickly it kills the flies. So there's no sort of trade-off between transmission and pathogen fitness, virulence and pathogen fitness. So yeah, and yeah, we're sort of still focusing on that middle step, and we haven't really looked into too much what will result in stain onward transmission in this system, because it's quite tricky to do.
And then another question from the same person was that viruses do not occur in isolation inside their hosts. So after the cross-species transmission, the virus needs to interact with the natural viral community already present in the novel host. How may this have affected the virulence and transmission potentials of SARS-CoV-2?
Okay. For SARS-CoV-2, I have no idea. But and maybe this person's, I think this is a great question, mainly because I've just put in, or just got accepted an extension to my fellowship on exactly this, trying to understand co-infections in sort of context of host shifts. So we know that things like rhinovirus infection is implicated in stopping the 2009 influenza outbreak in Europe. Herpes simplex virus 2 is involved in massively increased rates of acquisition, higher viral loads and higher onward transmission of HIV to HIV, and that's at like 40% prevalence in sub-Saharan Africa. So co-infecting pathogens can have sort of massive outcomes. For our own system, we've got some currently unpublished data using DCV and cricket paralysis virus. We've basically done single or co-infections of both of those viruses across host species. And the sort of take home, to cut a long story short, is there doesn't seem to be a consistent effect across host species. So generally, the sort of outcome of a host shift isn't really changed by the presence of the other virus in those cases. But they're just two viruses. I'm sure we're going to try with other different virus combinations. I'm sure we will find some effects there.
So Ben, there's a comment in the chat which you might want to come back to later on this, sort of following on from this. So you might want to follow up on that during the next talk. Right. So there's one more question from Su Sheng. So how does activity affect susceptibility, if at all?
I don't know what they mean by activity. Maybe they want to move.
Yeah. Do you want? Well, if you want to put up your hand, we can unmute you if you want to ask in a bit more detail what your question is. I should also have said, if people want to ask a question as well as putting it in the chat, they're welcome to raise a hand and we can unmute them. So David's actually got a question. David, do you want to ask your question yourself, or should I ask it for you?
You can ask it, David. Some human interaction's good.
Yeah, I'll ask it. So for the cricket paralysis virus experiment, obviously cricket paralysis virus is much further from DCV. So I was wondering whether or not you had any failures to manage to get infection in some of the species with cricket paralysis virus, and if so, was that phylogenetically correlated?
No failures. I think cricket replicates in all of the 45 host species. DCV replicates in all of them but one. Well, that's not quite true. We see a decrease in viral load in one of those species, but you can't tease apart whether it's replicating just at a lower rate than it's being cleared. So yeah, but yeah, cricket seems to replicate in everything, if I remember correctly.
Cool. Yeah, thank you.
I think we've got time for one more question. So Lena, have you've got your hand up if you want to unmute yourself.
Hi, Ben. That was a fantastic talk. It was so great to hear all of those things. I was really interested in the food part of the experiment. So you said that carbohydrates to peptides doesn't affect virulence at all, but that lipids might?
Well, we thought lipids might, but the pilot data isn't looking like, yeah, we can sort of massively increase the lipid content of the diet. The challenge with increasing lipids in the diet is you also increase calories. But we've tried to then do a sort of counter sort of control where we boost the calories, and yeah, we can't basically change the outcome of infection by mucking around with diet. We're doing this all in adults, so maybe the rearing diet is the thing to muck around with a bit as well, but then there's challenges with shifts in body size and things of the flies as well. So yeah, no, I mean, lipids seemed an obvious one given the sort of importance for sort of virus replication, but no obvious effects, though I haven't seen the final data set, but yeah.
Cool, excellent, thank you very much.
Great, thank you, and thank you, Ben, again. I'll just give you a round of applause because no one else can. For a fantastic talk. I should say that we've got a question and answer session at the end of the day, so